pull up a chair, take out the oxygen, and join the discussion.
You've just stepped into Life Unculturable,
the world where conversations grow faster than colonies.
We're your hosts, Sam and Julia, scientists by day,
podcasters by night,
and professional oxygen avoiders 24/7. Join us in exploring the science of microbes that thrive in the absence of oxygen on Life Unculturable.
We are live.
We are live for the first transatlantic edition of Life Unculturable.
Here we go.
So we're now the number one transatlantic anaerobic microbiology podcast. You know,
we're getting more and more specific. The number one transatlantic anaerobic microbiology podcast that is hosted by two guys and a girl.
You know, that's at some point you're not lying if it's that specific. So I feel okay with that.
Nice.
I feel okay with that. Yeah. Well, Julia, I suppose for those that aren't aware, you have recently vacated the United States in favor of Austria once more. And you know, how is life back in the motherland?
It's going. I've taken the public transport multiple times.
No.
Enjoying the pleasures of being able to walk around the city, bike around the city.
Hey, there you go.
Take a public transport. Enjoyed the mountains, which is also a thing that is non-existent in Oklahoma.
I was gonna say you need a little bit more west.
But I do miss the warmth and the sun.
Well, you can come back in August, and it will be permeating lava onto the surface of our great state. Man, so you haven't purchased a Herbie, then I take it.
No.
You're no longer in need of a little white Ford.
No longer in need of a very loyal and fun companion, unfortunately. But I am in need of a two-wheeled companion very soon.
Ooh.
So I'm gonna purchase a Herbie Jr., I suppose.
Oh, okay.
Yeah.
Self-propelled or motorized?
Yeah, it's going to be muscles all the way.
Muscles all the way.
Because electric bikes are kind of frowned upon.
Oh.
It's not cool enough to have an electric bike.
Oh, okay.
So I'm going to go with a self-propelled one.
Oh, okay. Well, that's good, because I was sort of thinking, gas-consuming motorcycles, so that's probably
of course, your American mind is going to the gas-consuming one.
Oh, yeesh. But I will say that, the price of gas, it makes me consider alternative options at this point. I think.
Where are we at in Oklahoma?
Geez, it's $4 and some change for a gallon. So I think when I filled up my truck last week, it was almost $100.
Wow.
Because I've got a 24 gallon tank, and I was like,
that hurts.
So that was tough. That was tough. I won't lie. Woo. Not gonna lie.
The rest of the world will just play a little song on the smallest violin for you.
Yeah.
Because that is still so cheap compared to the rest of the world. Because I think we pay. It's been up to €2 a liter, and there's 3.78 liters in a gallon.
Right, you are. No, that would be, yeah, because that'd be like what, €8 a gallon. Yeah, that would be tough.
I think it's not only Europe that's gonna laugh at you for complaining about $4, because before I fled the U.S., I was in Wisconsin and then Michigan, and you have one state has $5 a gallon, and the other one is 6 and some change a gallon. And then California is probably way higher.
Yeah. Well, I commend you on your anatomically, physiologically powered two-wheel mobile. So
powered by Schnitzel and Schokokuchen. Chocolate cake for the non-German speaking ones.
We go. That's what we need.
And maybe some bread.
Some real bread. There we go.
Some real bread.
That I would buy into. That seems like a product I can get behind. Man. Well, do we start out? Do we still have your signature welcome? Do we still get that?
Oh,
This is like your trademarked phrase if you needed to.
it's going to change now that I'm Europe. I need another
Yeah,
one.
or if it was going to
It's going to
change.
be Hallo und herzlich willkommen.
Hey, there we go. I will refer to a translator tool for that one. So. Oh man.
Well, Sam, what are we going to talk about today?
Oh, what are we going to talk about? I thought we were going to celebrate with schnitzel.
I would so love to like have you all over and like present you with all my favorite Austrian dishes. That would be a lot of fun.
Yeah. So I think, Julia, if I ramble things around in my brain far enough, we've been taking our listeners through most of what we do in the wet lab, a lot of the experimental things that we do, sort of from sample collection all the way through, trying to figure out what the optimal pH of an isolate is. And I thought sort of a natural continuum from there would be to take us down the unfortunate road of, what does it look like data collection wise? How do we process our data? And then ultimately in an episode or two, I think we start cracking open the Pandora's box of writing. And so I will say as a teaser, my lovely wife, Holly, so shout out, babe, if you're listening. I love you. She was like, oh, you guys should talk about what it was like to write your very first publications. And I was like, having a stroke in the car on the way to the appointment. And I'm like, can we talk about anything else? Like, is that an option? And, but anywho, so I think maybe it would be nice, as we sort of wrap up the wet lab, we involve the dry lab a little bit. We talk data, we talk informatics, and then I think we hop into the writing process. So I think today I would love to chat about sort of data collection and how we do and why we do it.
Nice. Yeah, I think that makes a lot of sense. And even though thinking about my first publication gives me like immediate post-traumatic stress. I think we can manage if I can like mentally prepare before we talk about that.
Yeah, we might.
We're golden.
We might need some time off for that one. I think I have to go sit in the mountain for a few days and meditate on.
Inhale grass. Not just touch it.
Oh, not too much because the listeners can probably tell I might sound a little bit off. I think I've got some sort of pollen.
Oh no.
I don't know. Whatever sinus stuff going on.
I'm sorry to hear it.
Oh no, that's okay. So if you tuned in for my voice, maybe you can tune back in next week, and it'll sound a little bit better. But yeah, that sort of had me thinking because we talk a lot about growing microbes, isolating and enriching and stuff like that. And I think when people think data analysis, they typically think like p-values and they think, really eloquently presented figures and stuff like that. And so the question that I wanted to open up with is, what do you think actually counts as data collection for us in anaerobic microbiology? For me, I think that data collection can be a number of different things. Some more glamorous than others, right? Like we obviously do a lot of things in like a replicate fashion, right? Because like good science is replicable and repeatable. And so as we talked about characterizing isolates, right? Like we do those things, we take those data down, whether we're recording optical density or colony forming units, which some of our guests may or may not like, which is fine. Thank you, Mel. Or we're taking pressure readings, right? Like those are data that we collect. We obviously collect metadata from samples and, locations like the age of something or the GPS coordinates or, that type of stuff. But I think some things that would be even overlooked are like, say if we're running like a qPCR, right? Like we'll collect data on like CQ values or, things like that. Concentrations of stuff in, say, a qubit. I think for me, all of those types of things go into data collection. If I was to try to surmise it and funnel it down, What do you think?
Yeah.
What comes to mind?
Yeah, I think it really depends on what kind of experiment or what kind of thing we're doing in the lab as to what pertains to data collection. And I don't think anaerobic microbiology data collection is that different from any other data collection. Like other labs will record optical density as well for their aerobic cultures probably. Other labs are doing qPCR, PCR, qubit, any of that sort. And other labs are also doing it in triplicates maybe, or technical replicates, whatever. So yeah, it just depends. Which our good friend Mel would call the standard answer. Oh, I think we cut that out of the episode, but I love that one.
Yeah.
I think her standard answer was it depends on the strain, because everything was just so variable depending on the strain. In this case, it depends on the experiment.
Yeah, I like that. I like the standard answer. No, that was really good.
Yeah.
There were a lot of nice little sound bites from that one. So if you haven't listened to that episode already, you should go back and check out the Melanie Eldridge episode. That was a really fun.
Yeah,
that was a really fun.
she's hilarious.
Yeah. Unlike the hosts of said podcast, very fun to listen to. Very hilarious.
One of us is.
And one of us is in Oklahoma. So, I like how you put that it's not just us as anaerobic microbiologists that are collecting data or even collecting data in this fashion. But I think that one of the best parts about science for me is the fact that there are sort of standards that transcend the disciplines, that transcend the fields. And that is like the need for, again, like replication. Like that's a big thing. Like, I think that's a really critical aspect that maybe some people miss, is it's not just sort of on a whim. Like things may come as interesting to us on a whim and we're like, whoa. But then you delve further into that and you figure out that it's repeatable. And that's I think a really cool thing about the scientific process.
Yes. And just one thing that just oh, I'm sorry, Sam. I didn't
No, go ahead. I was
you.
going to launch into a different thought, so you go right ahead.
Oh, yeah. I mean, one thing that just popped into my mind, and that was because I just like today interacted with some students, and I don't know how it was for you, but for me, I remember distinctly like the couple of first experiments that I did on my own in my master thesis, that there's a learning curve to actually collecting data, which sounds really stupid because it sounds like, hey, you get like this data off the machine, you write it down, and that's your data collection process. But there is actually a little bit of a learning curve because for some experiments you like have to note down multiple different things, multiple little things, especially in anaerobic microbiology, I feel, that you won't think of in the beginning. Like you start an experiment, if it's your first experiment, you think, okay, I just need the OD value, so the optical density value. But the more you do experiments, you realize, sometimes it could also be helpful to note down, I don't know, like how many hours they've been growing. So you know the next time you do the same experiment, because you often have to repeat experiments, you know exactly what your time frame is, like how long your strains took to like get to OD, whatever you wanted it to be.
Mm-hmm.
And so, yeah, that's just the mindfulness of like actually meticulously noting down every little thing to make your life easier in the future is what I learned specifically during my master thesis.
Yeah, I couldn't agree more. I think that for me, like you said, meticulously taking down all of those little notes about all of the sort of circumstantial things that are around you, like what was the pH of the thing, at what time, how long did it take me to get it transferred and then back in an incubator, was there a wait time there, how did I deal with the syringes, because it took me 15 minutes to do this the first time. And so I do find that if I go back and I travel back to like my first notes on doing something, they tend to be a lot better than my notes do like the 20th time that I do that thing.
Yes.
Because I begin to remember like, oh, I have to do it this way for it to be ready 30 minutes from now when I need to make this inoculation. And yeah, so I totally agree with that. And you sort of, over time, it's like you've built up that muscle.
Yes.
Which is perhaps why starting a new type of experiment is sometimes a pain. You're like, oh, not again. I don't want to. What do you mean optimize? Who optimizes? What is this crap? You know, like, what the heck? So, that's outburst number 1 for those of you that are counting. So, that's
fine. You don't have, just let it out.
Just let it out.
We love every facet of you. It's fine.
Oh, well, no, that's good. And speaking of facets, we've talked in the past that there's really 2 facets in the lab that we've, at least when you were here in Oklahoma. I get that you're currently involved in some different stuff there in Innsbruck. It is Innsbruck, right?
It is.
Not Vienna, it's Innsbruck. Okay.
I'm not so sure because I've been traveling too much.
I'm I got 2 guesses. There's only 2 cities in Austria I know, so it's roll the dice. Did I get it?
You did very well, and you actually pronounced it very well as well.
Oh,
so good job.
What can I say? Trust me, I'm a doctor. My wife hates it when I say that, but that's beside the point. Again, babe, if you're listening, I love you. But here in the El Shahed Yusuf lab, the 2 big things that we did were culture dependent and then culture independent work.
Yes.
And I think that the data analysis, the data collection process is vastly different depending on sort of which of those are we doing. Mm-hmm. We're not going to be doing pico-A plots if we're trying to grow something,
yes.
But we might if we're trying to, talk about ASVs or something like that in, a microbiome type data analysis. Mm-hmm. And so the question that I wanted to ask is sort of what is the difference between these 2 approaches, culture dependent and culture independent? And the more that I thought about it, the more I think that it's the level of bioinformatics needed, right?
Yeah.
Because I think if you're just talking culture dependent, I think you could get away with an iPhone calculator.
And maybe an Excel.
Or an Excel spreadsheet.
I mean, you can also draw it by hand on one of those older, those graphical papers with the little boxes.
Yeah. I think you could get away with that. Yeah. But I don't think that's the case if we're talking culture independent.
Probably not.
Yeah.
Probably not, yeah. So for our listeners, maybe the name already implies what the big difference is. One is dependent on actually culturing organisms, in the lab, and the other one is independent of that. We don't need the actual isolate or the actual culture. We could just there are different methods of looking at environmental samples without having to culture anything. So that's the main difference, I guess.
Yeah.
Sorry.
Oh, go ahead.
Oh, I don't think there was anything after end. That's the main difference.
Oh.
Period. Sorry.
Yeah, I was just going to say, I completely agree with Julia. Again, the culture dependent is just growing stuff. The independent is usually us looking at DNA or RNA, some sort of genomic information, whether that's all the DNA, a metagenome or a metatranscriptome, or maybe it's an amplicon survey, or it's just a specific gene or a specific chunk of DNA in an environment, but it's some sort of genomic content. I don't want to give the impression that culture-dependent stuff, growing things, is totally outside the realm of bioinformatics, because we totally do.
Oh yeah.
Totally do. We will sequence genomes, and we annotate those genomes, and that is obviously not happening with a TI-89. It's not happening with an iPhone calculator yet. So somebody out there, if you want to build an app that annotates my genome and does stuff for me,
on an iPhone,
send me
that would be
the
hilarious.
PM, my guy. Let's go. I would love that. But
But it depends on you having the culture first.
Yeah, you got to
not only the culture, but the isolate.
Yes. Yes, but even then, we're still, delving into, what is it? What are its cazymes? What antibiotic resistance genes does it have?
That's true.
Can we predict like its optimal pH with this phenotypic prediction tool? Like, so there's still things like that happen in the background. But yeah, I would say those are probably for me in a nutshell the differences, or the amount of bioinformatics.
Yeah.
So.
Yeah, and with culture dependent work, you can kind of sort of get around bioinformatics if you just don't try to predict anything and just test everything brute force. And then have the genome be sequenced and assembled by a different company and just attach it without doing any like analysis work on it. So you can sort of get around bioinformatics too, if you really try.
That's funny. See, I thought you were going to say you would then rope in like a co-author and just slap them somewhere in the middle, and you go, "Hey, do this really important thing, but just
don't tell me how, and don't involve me. Just give me the results."
Exactly. Just give me the results. Tell me when it's done.
Yeah. Well. So. How convenient, because I think there's like this really cool core facility at the Oklahoma State University called MCCF.
Oh yeah.
That kind of can help you with this part of your analysis, right?
No, I think I've heard of them. So yeah, for those of you that haven't heard me say this already,
not this episode yet.
I do belong. So lovingly, yeah. This will be the first time in this episode. I should get paid like per ad spot here, like the MCCF should.
There's a swear jar. There's a swear jar. There's like just the MCCF jar. $1 per mention.
Exactly. But I do so lovingly belong to the MCCF, so that's the Microbiomics and Culturomics core facility here at Oklahoma State. And like Julia said, that is essentially what we do. We sort of operate at the interface of growing things and computationally working on things. And so, like for instance, right now we're in the middle of a project looking at mouse fecal microbiomes. And I think we're somewhere in the neighborhood of 1,200 samples, I think. Ballpark.
Wow.
It's around 1,000. I don't know the specifics, but I can tell you that the computational time involved in running some of these pipelines is woo.
Even on the supercomputer.
It's small. Exactly. Yeah, the supercomputer is like, hey, slow down. We have other people at this university that are doing important things. On that note, send your boy some samples. Be sure to include your metadata. That'll be nice too. We love it when a client has nicely curated metadata. Yeah.
Nice.
And
so I guess for those of you that aren't necessarily familiar with the idea of bioinformatics, I think, not that I'm qualified to define this by any stretch of the imagination, but I think that bioinformatics to me, I would say, it's essentially manipulating biological data like in a computer in some fashion. And so I think bioinformatics to me, it feels simultaneously magical and also cursed.
Yes. Yes. 100%.
I don't really know how you feel about that, but
I so agree.
I think that there are so many incredible things that we can do using these bioinformatic tools that we possess, but I just don't possess the computational wherewithal to really squeeze enough juice out of this lemon as I probably could.
Yes.
Yeah, and so
it also, I think it kind of goes back to what we discussed in one of the other episodes. It's like, as a researcher, you're kind of supposed to be the jack of all trades, and like have all the knowledge about all the different fields, and you just don't. So you kind of figure it out as much as you need to, and all the like really deep dives, if you don't need them, you don't do them because there's no time or capacity for that.
Yes, I think that's very well said. Yeah, because I sometimes look at the figures that people generate and I go, wow, that is beautiful. Mm-hmm. And then I'm like, that would take me 16 hours and it still wouldn't happen. And it would be black and white. It wouldn't have any color.
That would be accessible for colorblind people, is what that is.
Yeah. Yeah. No, I am very well familiar with colorblind palettes in RStudio.
How so?
One of my co-PIs, he was colorblind.
Oh, really?
So one of my big critiques at the beginning was like, Sam, you've got to use better colors, buddy. Like, I literally can't see these things. I'm like, whoops. So my bad. But yeah, I think to me it's magical and it's cursed because we can get so much extra information, or we can make all of these really cool predictions using these data. But at the same time, it's like, those of you that are familiar with computer coding know, like, if you miss like a space, or if you hyphenate something instead of not hyphenating it, or you put two periods, or the wrong file extension, or there's a little minute error. There is no room for little minute errors.
Yeah.
And so you ship your code off to the supercomputer and it just goes failed, and you're like, what? And then you try it again and it's failed, and you're like, oh, okay. And then failed, and then you're like, how about a lunch break? That'll fix everything. And it
also, the supercomputer is very direct about that. Like, you literally get an email that says failed in all caps, like screaming at you. There's no softening the blow.
No, there is not. There is not. And I think you make a good point that, we're supposed to be sort of in a way these jack of all trades. And in bioinformatics, it's not that it's brand new to microbiology or anything like that, but I think it's becoming more paramount. You as a researcher are able to do some of those computational tasks that you probably weren't really expected or required to do in years past.
Yes, absolutely.
So,
but there's like different, I feel like every field, with microbiology, bioinformatics is like almost mandatory at this point, some level of it at least, in each field you're working on. Although when I did my master thesis, what I didn't expect that I had to learn was graphics programs, like Corel Draw and all of that, because of the like putting together Western blots and like putting the images together for the publication was like, Word couldn't handle all those big files, or PowerPoint, so you had to like do it in a proper program where you were able to open big files and then align them very properly and all of that. So I think each field has their computer stuff that you kind of have to learn, even though you're technically learning how to do the lab stuff in your studies.
Yes, and I know during my PhD sometimes people would say like, "Oh yeah, Sam, that's just something that you can do at home."
Yeah.
And I'm like, "What? When do you sleep?"
Never.
Like,
when you're dead.
Like,
or burned out.
Yeah, exactly. Like if I spend all day in the wet lab taking these time points for this transcriptomic thing that I'm trying to do, and then I go home and I manage to like eat dinner and acknowledge the existence of my wife and my children, and then I go, okay, Daddy's got to figure out, salmon and DD-seq2 to like process this RNA-seq data. And, at that point, I think I got like 5.5 winks of sleep, maybe. And so that to me, I don't want to say it was laughable, and I'm not saying that you shouldn't, learn things extracurricularly at home or, stuff like that. But I think to get to a level of proficiency in, whatever bioinformatics pipeline or computational thing you're trying to do, I think for me at least, it took probably more time than it should, because I'm not necessarily certain that my brain is wired to think in shell scripts and sort of ones and zeros and things of that sort. So that was a tough transition to go from like Migro dots on plates to banging out R code to like try to look at violin plots of some weird distribution thing.
But I feel like maybe newer generations of like researchers might have an easier time with that. I mean, of course, with the advancement of AI and all, but also because the curricula are made to include that. Like during my master studies, I purposefully picked like bioinformatic stuff as my electoral courses. I don't know if that's the same in the US, that you have like a couple of ECTS points that you need to do electorally. Like they're not mandatory. You can just pick what you want. And I picked bioinformatics because I knew it's going to be important. But the curriculum itself was not made to include that mandatory wise. And I feel like newer curricula might, because those curricula are also like, they lack behind. Like it takes a while to build a new one. It depends on the professors that can teach the course. Like, yeah, all of those little things.
Yeah, no, you're spot on, Julia. I think for me, like I recognized that in my master's that it was going to be a thing, whether I liked it or not. And it was sort of pitched to me in this pie in the sky way of like, oh, if you know how to do like a student's t-test in R, you're going to be, 5,000% more hireable in the future. And so I'm like, oh, okay. Like, all right, I'll figure out how to do this student's t-test in R. Kruskal Wallace, come on, buddy, let's go. And that's about as far as we took this wagon. And so I took one sort of elective course like that in my master's, and then I wound up taking a couple for my PhD, and they were all horrible. None of them went well.
Yeah.
And I mean, I suffered through and I made it, but like my confidence level in like telling another person how to do that is not very high. Like I could turn someone into, a person that cultures obligate anaerobes from whatever environment, but I don't think I could turn you into like an R expert or a Python expert or, pick your Perl, whatever language you want to do this in.
Yes.
I don't think that's me. And I started collecting like a file full of little scripts that worked.
Yeah.
And it's like, this is the script to do like a protein blast or a nucleotide blast, or this is the script that runs this, phylogenomic tree pipeline, and this is the RaxML script, and this is that script. This is Prodigal, and so I have a file to this day that just in all caps it says Linux help. And and your boy goes back to that frequently because I need the helps.
Yes.
And I'm like, if it ain't broke, don't fix it. And then it gets broke, and I don't know how to fix it.
Because then the update comes, or this package gets discontinued, or
No, that's a tough one.
Yeah.
So it's hard for my brain to shift gears and to think about stuff like that. It's a very difficult thing. I think I'm slightly better at it now, but not much better.
I think it's also hard, like when you've made little progress and it's like all baby steps from 1 minute to the next. It's hard for you to recognize how much progress you made from the beginning of your PhD to now. So I would say probably like genome assembly. I would go to you and ask like, how are you assembling your genome? And you would probably be able to answer that. If I'd ask first year PhD or second year PhD Sam, he might just look at me with like question marks in his eyes.
Oh, I still have those.
So I think you need to give yourself some credit here for the progress you made. But
no, that's fair.
And also with bioinformatics, I feel like it's even more like with the culturing stuff and all the lab stuff, you also don't really realize the huge progress you make over time because you do it in such little increments. But I feel like with bioinformatics, that's even worse. Like that is factor 1,000.
Yeah.
With like the increments are so small, and each time you do a data analysis, you learn like this little thing and then this little thing, and it feels horrible all the time. So you don't recognize the progress.
Feels horrible all the time. So I think we found the name for this episode. It feels horrible all the time. Oh boy. So yeah, that took. I will tell you that there's nothing that makes you feel more defeated, that made me feel more defeated than struggling over the weekend trying to get some sort of code thing to work, and it would like half work or 3/4 work, but it didn't work. And then you'd go into your supervisor's office and you'd be like, "Hey, boss man." I would lay it out like, "Oh, I've tried this, that, and the other thing. Like, oh, the sed with this awk, and I tried to do and pipe it into this, and I'm having trouble." And that man, he would just look down at his computer, and it would just be like, bing, and it would just shoot out the thing, and I would be like, "What?" Like, it would just take like a minute, maybe 3. And then I would just stare at the screen and I would be like, "Please don't ask me how long I tried to do this for. Like I was literally trying for like 15 hours and you fixed it in 3 minutes." I don't know if that's ever happened to any of you or our listeners out there, but the struggle is real, folks.
It is. It is real.
That was a rather defeating moment I remember early in my studies, as being like, I don't know how he's such a wizard. What is going on here? So that was tough.
But it's good to know that there's like somebody who can figure it out in case it doesn't work for you.
Yes. Yeah, I don't look forward to being that person.
Because there is... Yeah. I was about to say there's like nothing more humbling than having a problem and then having nobody to immediately go to fix that problem, and you realizing you are the one supposed to fix this problem.
Yes.
That is tough.
Yeah.
That requires the fetal pose and some crying and maybe some chocolate ice cream for a while, and then...
Yes. Yeah, it's funny you say that. That's almost exactly what I tell people, and so we had a student in the lab the other day, a trainee, and she was getting some, we'll call it anaerobic microbiology 101 from yours truly. And things just were not going according to plan. And so at one point in time, I looked at her and I said, somewhat jokingly, I'm like, "If this doesn't work, this is when we go to Brahms and we cry and we eat ice cream." I don't know what else to tell you, but that's the part of the curve that I'm at. And I'm like, if this one doesn't work, we're gonna. I'm gonna have to brainstorm this for a minute.
Yes.
And so those moments are very real. But yeah, I think even more so in a computational sort of data analysis type way for me, because it's like, well, we're just gonna close this computer now and sulk into the couch, and
yes,
we'll revisit it next month, maybe. So, yeah.
But that sometimes is the solution. Sometimes like closing the computer and coming back to it sometime later is like with a clear mind. Sometimes just rewires whatever knots were forming in your brain.
Yeah.
And you're good to go.
Yeah, and I think too, even that in in a wet lab sense, because I know for a while during my PhD I was doing like several late nights in a row, and it was coming to a point where I would be at school, say your typical 9 to 5, like come home, have dinner, put the kids to bed, and then I would drive back to the lab, and then I would be there until past midnight, 1 o'clock, 2 o'clock, like that sort of thing rolls around. And then you're looking at your watch and you go, "Man, Sam, you got to be awake in, 4 hours, and you've still got a 30-minute drive home." And and there came a point where like I told myself, I'm like, the work that you're probably doing now is probably of not the best quality, because you're half delirious. You've just been snoozing on like the whatever nth edition of the prokaryotes, because there's nowhere else to sleep in this lab. And it was like, okay, I can do like maybe 2 of these a week. Like you can't do like 4 of these back to back to back. Like you just can't do it.
No.
So I agree with you. I think sometimes time away, even though it feels wrong and it feels like you're not doing the right thing, is probably the right move and the best thing you could do.
Yes, absolutely.
Yeah. Back to data sets, Julia.
Yes.
I was curious if you thought that there were common ways that we, the royal we here, sometimes ruin our own data sets.
Oh my.
Because we sort of talked earlier about, bioinformatics pipelines and things needing to be structured a certain way and all of the file extensions and stuff like that. And so I'm curious if you have an opinion on ruined data sets.
Boy, do I have an opinion on that. It never happened to us, of course. We are perfect in every way. I think one of the most common ways that you can make a data set horrible is, like, say you have a data set either of multiple samples or multiple time points, and then you record different things at those time points or for each sample. For example, you have age, sex, feed, location, and let's say you don't record all of those for all the samples. I think it's the most common way to ruin a data set is by having it be incomplete. Like sometimes you can't get around that. Sometimes, there's just not a data point for each one of those. But you should always try as best you can to have a complete data set.
That seems fair.
That is Datasets 101.
Datasets 101. Well, I would add to that in the second semester class in Datasets 102 that you should probably input your data in a similar fashion throughout the data set.
Oh, yes.
So I've seen some where, the person will start out writing like, sample 001, and then a few lines down, it's sample 19. And I'm like, if we're going to do the whole 00 thing, like if you've got 001, you should probably have 019.
Yes.
And little things like that. Like I've seen some where some lines have like spaces, but some lines will have underscores and different things like that. Because when we go to split data sometimes within a column or within a row or something of that sort, we'll look for a character, whether it's, again, an underscore or a colon. But I think as far as data sets go, it's important to, at least it's important to me, to try to maintain sort of the integrity of what's in the columns or the rows, depending on how the data set's oriented. Because nothing is worse than getting, in my opinion, like a data set from a collaborator who shall remain nameless. And you're like, what is this? There's like 87 columns and none of them make any sense. And they're all formatted differently. And that's tough. And also your sample IDs. I feel like if you give your sample ID some sort of a marker, it should be fairly intuitive.
Yes, and ideally.
It should make some kind of symbolic sense.
Yes.
Which it often does not.
Yeah.
In my experience.
Nice.
And
101 and 102 data sets. Yeah.
Yeah, and that's probably where that course should stop. Like you should just get a certificate after 102 and not learn anything else.
Yes.
So
there's nothing else to learn.
I'm with you on that. Or I will say another thing too that kind of irks me is when you have the same sample but it's given a different label.
Oh, yes.
So like maybe you have been working on this particular facet, your collaborator's been working on a different facet.
Yes.
They're collecting a certain type of data, you're collecting a certain type of data, but on the same thing.
Yes.
And then you've named the same thing wrong.
Yeah.
Or differently. I guess wrong is the not appropriate word here, but theirs is obviously wrong. And so that's tough at times.
Completely agree. But I think to summarize, it's like keep your data sets organized is like the main thing, right? Mm-hmm. Like have your samples have one name throughout the data set. Like each sample has a very unique name. Keep it organized. Keep it short and sweet. Like no sample needs like a whole poem or paragraph worth of a name. Like a couple of letters, maybe a couple of numbers, we're good. Like no matter how many samples you have in there, I don't think there's any reason for a sample name to have more than 3 numbers and 3 letters or something like that.
That was interesting. I was just about to ask you, like, what's the upper limit on characters for a simple name? Like, are we over or under 10? And I was like, it has to be under 10. Like, it's got to be under 10. Like 10 is.
I mean, if you have very, very large data sets, you might want to pick like 1 number more or 1 letter more, just to make sure you have enough combinations of those numbers and letters.
Yeah.
But even then, like 3 and 3 gets you far.
True. That does get you pretty far.
Yeah.
And then, Julia, I suppose the last thing, again, assuming we survive this, is once we have all of this perfectly curated data and it's been professionally analyzed by somebody other than me in our studio, what in the world do we do with that next? Like, what do you, in your mind, you're thinking, oh, we've generated all this data. Now we've got it in a format that we like. Like, what's the next step in this process?
Put it in a PDF file and hang it on the fridge after you print it in.
Material.
No.
Don't waste that PDF on the fridge, kids. Bury that PDF in the supplemental material of your next manuscript. See a previous lab yap for that.
No, so ideally, of course, once we have all of this data, I mean, you're talking about already analyzed data, or are we talking just the curated data set?
No, I'm thinking analyzed. It's ready to
everything.
Go. Yeah, we're totally.
You have like pretty figures and all of it?
Oh, yeah. With figure 1 through
nice.
S2E is constructed.
Well, then we hopefully construct a manuscript around it and submit that sucker to our favorite journal.
Yeah.
Right?
Yeah. No, that's...
Well, because if you have the perfect analyzed data, it's curated, you have the figures, you just have to write the manuscript around it and then submit the manuscript, the figures, and ideally your curated data set in a manageable format to a journal to be peer reviewed and published. Hopefully.
Okay. So I think what I'm hearing here, folks, and what you're hearing here on Life Unculturable, is that we should probably talk about manuscript drafting, perhaps the writing process, how we write, how people shouldn't write, and I think even the submission process, like peer review as a whole, that could probably be talked about.
Yes.
We can leave off the APCs and we can save that for a yapisode in the future.
But also the accessibility of like curated data, like how do you make it accessible for other people? Could be another interesting talk.
That's very true. Yeah.
Open access, open science.
Yeah, open access that's a nice thing. Data availability is also a nice thing. I know that was one of the things I picked on in a lab yet previously, is when you have to like email one of the authors for the data. It's like available upon request, and insert that yap here, folks, because I still feel the same about it. Curse you. Yeah, I don't get it.
That was a very mellow expression.
Well, I'm trying to remain hireable. So one of us is no longer in that spot, and the other one is, Well, Julia, I was thinking that it might be time for us to dive into some questions from our illustrious undergrads.
Yes.
How do you feel about taking a swing at some of those?
Let's go. I love the questions.
And I will say, I'll preface this by saying that we have recently lost, not to death, but perhaps they're dead to us now. Some undergrads who have moved on to bigger and better things, med school and graduate school and all those type of things. And so before they left the lab, which was just this last week, I had to make sure to snag a last round of questions from them. So they've graced our presence with three questions. I'm not really sure. I'm staring at them now, and I'm like, well, I'm kind of scared to answer two of these, but the first one that I'm looking at says, if bioinformatics pipelines had Yelp reviews, what would yours say? Oh,
2 stars. Don't recommend using.
And yeah, the first thing that comes to mind is like, have you been to the restaurant across the street? Like, like why are you dining with us today? Like, take your business elsewhere. So I think my Yelp review would be something like, eventually gets it done, questionable at best, could
Needs a lot of RAM.
Yeah, needs needs a lot of something. So I think that's what mine would be. I probably wouldn't have 5 stars. I'd probably be lucky to have 2. I guess depending on the type of analysis we're talking about here. But yeah, I think that's probably what mine would say.
Yeah.
Do you think yours would have 5-star reviews or?
Oh hell no.
What would your Yelp review look like?
I think yeah. All the you said, plus maybe, needs a lot of computational power. And also only works under very specific versions of the different packages. It's like, has worked for version, 2001.4, but is no longer working in like version 2003.7.
Yeah.
Something like that.
Okay. So
works really well for this one very specific analysis, doesn't work for anything else.
Yeah, that's a great point. I'm thinking that I would have to be like one of those restaurants where, they serve like 5 dishes and that's it.
Yes.
There's no customization.
No.
You're getting the steak exactly how I like to cook it and all of that stuff. Like that would have to be my Yelp review. Be like, it does 5 things very well.
Yes. Yeah.
No extra fluff.
No substitutions.
Yeah.
Nothing.
Yeah, no. No customization, no substitutions.
Yeah.
Yeah.
Okay.
Okay.
So we got the Yelp reviews.
Yes, we got in our astounding Yelp reviews. And on the subject of Yelp reviews, I see the next one is dating profiles. So,
oh boy.
Yeah, I'm not going to guess which undergrad put this one in, but I have suspicions.
You can tell that it's springtime, right?
Spring has sprung. Let's see. So it says, if anaerobes made a dating app profile, what would their bio say? So I'm imagining, you're swiping through, and then you just see some anaerobes pop up. What might an anaerobe put in their dating profile? And I guess this could be 2 parts, right? Like, what should an anaerobe put in their dating profile, but what would they actually put in that profile? So I'm gonna let this roll with what would they put in their profile?
It depends on the anaerobe, I guess. If you like to breathe oxygen, swipe left. Probably in there. And if you can't handle all my secondary metabolites, swipe left.
Okay. For those of us that have been married going on 9 years now that didn't get to quite experience that revolution, I'm presuming left is the not.
It's a no.
Right. Okay.
Yes.
Okay. Perfect. I think an anaerobe would probably have like a list of specifications for the person that they're looking for. They'll be like, you must be this, that, and the other thing. It's like, must be able to tolerate my nonsense, must be able to keep me at very specific temperatures. I only like certain atmospheric mixes. My atmosphere must contain CO2. I think.
I like how we both went the toxic route, because I feel like dating profiles that only list off things that the other person needs to have instead of things about the person that the dating profile belongs to is like already a red flag. It's like if you have a list of demand instead of like introducing yourself, red flag. So I feel like it's very funny that we both went the like kind of toxic route.
But I'm trying to think of like what's the upside? what's the upside?
In dating apps these days, you can like you have different categories of characteristics you can choose from. Like in like hobbies, you can have like hiking or yoga or movies or whatever, and then you have like supports Black Lives Matter or is vegan or something like that. I feel like in all of this, there would be a lot of very specific nutritional needs, like maybe not vegan, because we talked about that in the other episode with like the brains, hearts, and bloods infusions, but like carnivorous is maybe the thing.
Yeah, there we go. Yeah. Like I only eat at restaurants that have 0.1% yeast extract. You know.
But then there's also like
volume rumen fluid. Take me to the slaughterhouse, baby. Like, what are we doing?
And then my filamentous fungi, they would probably go. I love cuddling because they like always clump together. So there would be something about like cuddle or.
I see that. Enjoys
something
long
like that
walks by the anaerobic digester. Will find me at the wastewater treatment plant in the omasium of a cow.
The spa day.
Okay.
I think there was an episode where we did like the holiday places for
Yes.
anaerobes. So, yeah.
Yes. Yeah, I think that's the one that just came out. Maybe. It's the one that I was listening to when I told you and Michael I was off to get donuts early in the morning. I think it was that one. But yeah. Okay. So they maybe anaerobes could have dating profiles, some more successfully than others.
Certainly.
Certainly those that are capable of coming into culture. Those will probably be the easier ones to get along with. And then the last question that our glorious undergrads so graciously gifted us is, why in the world do we become so emotionally attached to certain data? And I don't know whether to feel a sense of proudness about this or if I feel like I'm being attacked by people that are 10 years younger than
Like,
me.
what do you mean emotionally attached?
Yeah, I'm not emotionally attached. Like, what is this? So I feel I have mixed feelings about this.
But also, I mean, of course you are emotionally attached. You like invest a lot of time, and there's like what is the cognitive bias called? Where you're like, if you have invested a lot of time into something, you're more likely to positively rate that something. I forgot what the name of that bias was. So if you've like built your little sample collection Excel sheet, and then one undergrad comes along and like changes all the formatting in it, of course I'm going to have an emotional reaction about my precious little 2-year in the making Excel sheet.
Yeah, I was going to say we might have to have them meet an untimely end. We'll send them to sample from the ostriches. Yeah, that's tough. I think that's probably, time. You mentioned like, for my 2 years, like that's probably it. Is it's the amount of time that went into maintaining, a consistent data sheet or protocol, and I think sometimes some of our studies they come together over the course of months or even years. Yeah, so I think a lot of that for me is like the time that I've invested in a particular thing.
And not only that, then there's also like the mere exposure effect is I think what it's called. It's like you're more likely to like a person if you see them every day. So if you look at that data sheet every day or every week, I feel like that triggers the mere exposure effect in you.
Okay. That must be why I have all those sticky notes all over my desk. So I can be reminded of these. Don't do your data collection on sticky notes, kids. Like if you're listening to this, don't do as I say, not as I do. Let's put those in an Excel file, Google Sheets, LibreOffice Calc. Like
there we go.
Pick your spreadsheet, but don't put it on a sticky note.
Yeah.
I can't tell you the amount of times I've been like, oh babe, like there's this sticky note in the office and it's yellow and it's on top of this one. It's a different sticky note that's on this page. Like, can you take a picture of that? It has something to do with like a dilution that I'm making that day, which is in retrospect very important. But was on a sticky note in a different zip code. So there's that. Man, emotionally attached. Oh, and I think too there's this this sense of anticipation, and especially like the closer you get to being done with something, and you're like, okay, the plan is to submit this in, the end of April or the end of this month or before the end of the semester. And you're anticipating that, going into a particular journal, and you're like, oh, we need to format the supplemental material this way, or we need to get these tables so.
Yes.
And then I think you're a little bit excited about that. Cue the time thing, because you're like, oh, we spent the last 8 months working on this, and now it's finally in a place of, oh, it's in someone else's hands.
Yes.
And so I think that can be emotionally damaging or satisfying. It's a little bipolar.
Yeah.
It's a little bipolar.
Yeah.
That's probably what I would say. And maybe have copies of it in other places.
Yes.
Don't lose that data on a whim.
Back up your data.
Yeah, that's a rough one.
I feel like every researcher has this like very traumatizing event happening to them where they finally learn that like backing up their data is really important, and backing up the current state of the manuscript or the thesis or whatever you're writing. And then usually by the time they're at the PhD thesis writing stage,
Yeah.
I think most researchers would have like a million different files that are all just this one PhD thesis, because you're frantically like saving it on different folders, computers, external hard drives. But yeah,
that is so true. I tell you, that traumatizing experience for me was in my master's. At the time, my wife and I, we lived in this little apartment in Joplin, Missouri. And oh my goodness, there was barely a living room. And so I would lay in the hallway and just put like some pillows underneath me, and then I would just type and do my like stuff laying in the hallway. And that's how I would do most of my work. And I can remember it was like late at night type deal after, she'd went to bed. And it was like the computer just went a color that a computer is not supposed to go. Like your Microsoft Word, you're typing away, blah, blah, blah. And then it's like royal blue. And you're like, that's not supposed to be blue, And I thought my computer was fried. And in that moment, you're like in the semester of graduating, and you're trying to get all of these things done. And I'm not going to say that we got noise complaints that night, but I will say that I was upset. And then thankfully my wife, she got a degree in, her degree was in computer forensics. That's what she did, computer forensics and criminal justice.
Wow.
And so she was able to snag my data back from the brink of death.
Nice.
On that old computer. Thankfully, it wasn't all for naught, but I will never forget that feeling of I have royally screwed up. I need this in like 11 different places. And I don't know about you, Julia, but I know for me, like during my PhD, I would take my laptop with me everywhere.
Yes.
Even if I didn't need to. I'd be like driving to my mother-in-law's house on the weekend, and I'm not even going to work, but like I'm bringing that thing in the
just
bedroom
to keep an eye on it.
Like it's going everywhere. Yeah, it's like a nuclear football. I've got like a handcuff to my hand, a suitcase in my hand. Like it's not going anywhere. But also, I'm not going.
Yes, agreed.
Yeah, I couldn't let it out.
But also, like when I think it was one of our undergrads in Oklahoma State that I talked to of like backing up data, and they were like, no, it's all on the drive. Like it's all in the cloud. Nobody needs to back up their data anymore. Well. Recent traumatizing experience. I don't know if you remember that. I think it was episode 3 or something like that. I had a computer at home. I had the computer at work. And sometimes like when I'm done with all the work for the day, I would just either work on my computer at home or at the computer at work on our episodes, like editing those episodes. And I didn't know it back then that I need to wait before I shut down the computer because the OneDrive needs time to synchronize before the computer is shut down. I was like, it's on the drive. Like I have it saved on the drive. I close the file. It's immediately on the drive, in the cloud. all was fine.
Oh.
And it was like Wednesday evening before our Thursday morning episode drop day. And I realized all of the editing that I had done is gone.
Yeah.
Yeah.
I remember that.
That was tough.
Yeah.
But yeah, so when you do have a cloud or a drive system, that's nice, cool. Maybe still back up your data and also wait until it's synchronized before you shut down anything.
Yeah. Well,
And especially those audio files, like when you edit them, they're so huge, it just takes a while. So it's like you can have the fastest internet and it will still take a minute.
oh, this is quite the life advice episode. Oh, what a way to possibly end this.
Nice.
I think that was pretty good.
Yes.
No, I will say, Julia, it is lovely to be back in the podcast chamber with you.
Yes.
Even though you're setting somewhere, I presume, in Innsbruck. It's only one continent away, one ocean, half a continent, whatever. We'll round.
Yes.
It's fine. But it's been good. It's been nice to catch up, and I'm so glad that things are going well for you.
Aww.
And hopefully they continue to do so.
I really love that we continue to do this even across the Atlantic.
Yes.
And I'm really glad it works so well.
Yes.
So
Yes.
yeah, and I ideally miss you guys.
Yeah. Well, we certainly miss you. Certainly do miss you in the lab. But yeah, if you guys haven't already gotten tired of us and you would like to hear more from us, by all means, check us out online, as our internet presence is growing day by day. I just checked on LinkedIn yesterday, and I think we were at like 113, maybe something like that.
Wow.
So yeah, we were shooting for 100 for a while, and we're already 13 over that.
Nice.
And yeah, so you can check us out on LinkedIn, Instagram, Blue Sky, YouTube. Pretty much anywhere you listen to a podcast, Spotify, Apple Podcasts, all that stuff, Audible, we're all over the place.
We are.
And we look forward to being in your ears.
Well said.
So, yeah. And I suppose on that note, Julia, I will bid you adieu until next time.
Bye, Sam. Until next time. Looking forward to it.
All right.
And I'm going to say very Viennese, bye-bye.
Ah, that's
Is how we say bye-bye.
okay.
Bye-bye.
Man.
Bye-bye and Foynet.
Is that good night? Foynet? Is that no?
No,
I
it's
won't
just
try.
that's a long story what that is. It means bye-bye and don't hurt yourself,
Oh well.
basically.
I will try not to do that today. if you're the health and safety person and you're coming into the lab, I'm your boy today, so
Nice.
let's go.
Well,
All right.
bye everybody.
Yes. Bye, everybody.